In this chapter now, we're going to be looking at the third and final line of defense, which is the adaptive immunity. Just to remind you, we looked at the first and second, which were innate, non-specific, and you'll be looking at specific responses. So adaptive immunity is acquired only after an immunizing event. This immunizing event could be either an infection or a vaccination. So when is that going to happen? It's only when the host barriers and those innate defenses are failing to control an infectious agent. Then, when you have a person who has a normally functioning immune system, this third and final line of defense is going to kick in to resist the pathogen. So it's not innate, it's adaptive and acquired after an immunizing event. Adaptive immunity is a product of a dual system, which we had looked at earlier, which has the B and the T lymphocytes. During fetal and early postnatal development, you see a selective process that goes on with these lymphocytes, and they are prepared for reacting to a particular antigen, like it's very specific, for a particular antigen or immunogen. At this time, the immunocompetence, which is the ability of the body to react with countless foreign substances, is developed. So when a baby is born, they already have the theoretical potential to react to an extraordinary array of different substances. Now, antigens are molecules that can be seen and identified by the immune system. They may or may not provoke an immune response. So in order for them to provoke a response, they'll be sensed by the immune system. But even if they are sensed, they may or may not provoke a response. If they do provoke a response, they're called immunogens. Now, these molecules are usually protein or polysaccharide molecules, and they're either on or inside all cells and viruses, including our own cells. If you have different environmental chemicals, those can also serve as antigens. So any exposed or released protein or polysaccharide is potentially an antigen. Like I said, even those in our own cells. So our own cells normally do not provoke a response from our own immune system. And that's the way we want it. If there was a response being provoked, we would call that an autoimmune issue or autoimmune problem. And actually, the ability of a lymphocyte to discriminate between differences in molecular shape is so fine that it will recognize and respond to only a small portion of the antigen molecule. That antigen fragment is called the epitope, and that's the primary signal that tells the body that this molecule is foreign. Just a reminder, we had talked about PAMPs, pathogen-associated molecular patterns, in the previous chapter. Those are stimulating responses by phagocytic cells during an innate defense response. And remember, these are molecules that are shared by many different kinds of microbes. They will stimulate an innate response, whereas antigens are highly unique to individual microbes, and they stimulate adaptive immunity. So now looking at this infographic, we start to get a list of different characteristics that make adaptive immunity unique. Two of the most important ones are specificity and memory. So in general, when antibodies are produced during an infection, they will function only against that particular pathogen and not against other ones. This property of memory, that's the specificity. And then the property of memory is referring to the rapid mobilization of lymphocytes that have been programmed to recall their first engagement with the invader. So you're exposed either through vaccination or maybe an infection. You have memory cells that retain the programming. And then when you're re-exposed to that invader, they will rush to the attack again, which means that they will be revving up the engines and producing antibodies. So looking at other characteristics, specificity we looked at in memory, but in between there's also diversity. So there's always at least one cell that can react against any antigen. Inducibility, only turned on when triggered. Clonality, millions of cells can be cloned, so they would be identical with the same specificity. And tolerance, it's not going to react with self-antigens, unless, of course, there is an autoimmune issue. Now, the elegance and the complexity of the immune function is largely due to lymphocytes working so closely together with other leukocytes, like macrophages, dendritic cells, and the innate lymphoid cells. So in order for us to simplify and clarify this network of immunologic development and interaction, we're going to look at it as a series of stages, and then we'll cover each of those stages. So now it's really just introducing those 4 stages. The first is lymphocyte development, clonal deletion. The second is when the presentation of the antigen happens in this clonal selection. Challenge of the B and T lymphocytes by antigens. And then there's a T lymphocyte response, which we call the cell-mediated immunity. And there's the B lymphocyte response, which is where the production of antibodies happens and where all their activities go. If you look at the first stage, the lymphocyte development, although all those lymphocytes arise from the same basic stem cell, we find that at some point in development, they diverge into 2 distinct types, the B cells and the T cells. Final development of these B cells happens in specialized bone marrow sites, and the final development of the T cells happens in the thymus. Both cell types are then migrating to separate areas of lymphoid tissue, so different organs like the nodes, lymph nodes, or to the spleen, for example. B and T cells are constantly recirculating through the circulatory system and lymphatic system, so they migrate in and out of these lymphoid organs, constantly patrolling. Here you see an overview of this lymphocyte development and function that I was talking about. So here you have the common lymphoid precursor in the bone marrow, clonal deletion happening here, specific bone marrow sites and B cells developing. And here in the thymus, you see the T cell development happening. So all of this happens independently of antigens. This is happening regardless. Then there are the antigen-dependent responses, which we'll be looking at later. So we'll come back to this image and we'll look at this again. We've only talked about this part for now. So entrance and presentation of antigens in the clonal selection is going to be the next thing that we're looking at. When you have foreign cells like pathogens, which are carrying antigens, and they cross that first line of defense. Remember, that's your skin, your mucous membranes, etc. And then they enter into the tissue. You will have resident phagocytes that migrate to that site. The tissue macrophages will ingest the pathogen, and they will induce an inflammatory response in that particular tissue if it's appropriate. So phagocytosis happening there. Tissue dendritic cells ingest the antigen and they move to the nearest lymphoid organ. Typically, this will be the draining lymph node that is close to that. There, they're going to process the antigen. They'll present it to the T and B lymphocytes. And in most cases, the response of the B cells will require the assistance of T helper cells. T helper cells are a group of T cells, a special group of T cells that, as the name suggests, they help. Now, markers and receptors have their own roles to play. So all cells, whether they're foreign cells or cell cells, have a variety of different markers on their surfaces. Each of these, each type, has its own distinct and significant role that it plays in detection, in recognition, and in self-communication. Cell markers play really important roles in the immune response because they activate different components of immunity. And because of their importance in the immune response, we're going to look at the major markers of lymphocytes and macrophages. So the major histocompatibility complex, in this, there is a set of genes that codes for human cell markers or receptors. This gene complex produces a series of glycoproteins, which we call MHC molecules, and they are found on all cells except red blood cells. The major histocompatibility complex is also called the human leukocyte antigen system, the HLA system. This set of markers plays a vital role in recognition of self by the immune system, as well as in rejection of foreign tissue. Now, there are three classes of major histocompatibility complex genes that have been identified. Class 1 genes, these code for markers that appear on all nucleated cells. They display unique characteristics of self, and they allow for the recognition of self molecules and the regulation of immune responses. So this is a complex, complicated system when you look at all the details. But in general, what we're trying to say is that each human being inherits a particular combination of class 1 MHC genes. And this is pretty predictable. Now, class 2 MHC genes, they also code for immune markers. They are found in macrophages, dendritic cells, and B cells, and they are going to be involved in presenting antigens to T cells during cooperative immune reactions. Your class 3 MHC genes encode proteins that are involved with the complement system, amongst other things. And we will be going over the complement system a bit more. So here you have classes 1 and 2 of the major histocompatibility complex. On the left is an image of an MHC molecule, class 1, found on all nucleated human cells. And here you've got the antigen binding cleft. Here you have an antigen binding site, which is on a class 2 MHC found on some kinds of white blood cells, which has class 1 as well. Both of them you can see here. CD molecules, CD stands for cluster of differentiation. So these are really important markers in immunity. And when we say CD, this is basically just a naming scheme for many of the cell surface molecules. There are more than 400 different CD molecules that have been named, and many of them are involved in the immune response. Some of the important ones are CD3, CD4, and CD8, and you'll see their names coming up as we continue through this. Now, markers are lymphocyte receptors. So lymphocyte markers that are often called receptors. Both names are used, but the emphasis is that their major role is to accept or to grasp antigens in some form or another. So B cells have receptors that bind antigens, and T cells have receptors that only bind antigens that have been processed and complexed with MHC molecules on the presenting cell surface. A little difference between whether they can accept them just in their, I'm going to say, raw form, or whether they have to be presented in a particular way. There is an image in your book that actually presents the surfaces of T cells and B cells and demonstrates it pretty well, figure 13.3. So in that, you can see the surfaces of B and T cells, their antigen receptors, and various different kinds. So potentially there are millions or even billions of unique kinds of antigens. The many sources of antigens include microorganisms, cancer cells, as well as an endless array of chemical compounds. When challenged with an antigen, B cells and T cells, both of them proliferate and differentiate. So the multiplication of a particular lymphocyte creates a clone or a group of genetically identical cells, some of which are memory cells, and those will ensure future activity against that antigen. Because these B cells and T cell responses differ significantly, we're going to be looking at them separately from this point onwards. Now, how T cells respond to an antigen. You have three kinds of T cells. There's a huge variation in their types and their responses, and so we look at them individually. When they are sensitized by an antigen, they give rise to a variety of cell types, completely different roles. And the first one is the helper cells. We mentioned this earlier. They're helping the B cells. So they activate macrophages, assist B cell processes, and they help to activate cytotoxic T cells. The regulatory T cells, they regulate, so they control the T cell response by secreting anti-inflammatory cytokines, or they prevent proliferation. The last kind is cytotoxic T cells, cyto and toxic, meaning toxic to the cells. So this leads to destruction of infected host cells, as well as other foreign cells. T cells secrete cytokines that help destroy pathogens and regulate immune responses. They don't produce any antibodies. They just secrete cytokines that will help regulate immune responses as well as destroy pathogens. Recently discovered innate lymphoid cells, the ILCs, engage in parallel activities to the T cells when they're activated, but they don't use specific antigen receptors the way T cells do. Now, how do B cells respond to antigens? When they are activated or sensitized by an antigen, they divide. This gives rise to plasma cells, each with the same receptor profile. Plasma cells release antibodies that only recognize entities with that same antigen. The antibodies then disperse into the tissue and blood, and when they attach to the antigen for which they're specific, that antigen is then marked for destruction or for neutralization.